Hi, everyone. It's my distinct pleasure to introduce Professor Jack Feldman. Jack got his PhD in physics. Like a lot of the great neuroscientists here, they started as a physicist and then turned into neuroscientists. He did his PhD, he did his postdoc 1 year in Paris and then went to Einstein College of Medicine, and he stayed on as faculty. Then he moved to Northwestern, rise through the ranks as full professor. Then he was recruited away by UCLA, and he was at UCLA since. So Jack is very famous for his study of breathing. So in this landmark paper in 1991, Jack discovered the center, the location in the brainstem that generate this constant breathing rhythm that allow us to be alive, right? When you fall asleep, you don't think about it, and you still breathe. And so most people introduce Jack as the leading expert on brain control breathing. For me, I think Jack is the Pope of breathing. Okay, so I think you'll hear from the Pope today about breathing. He knows everything about breathing. For his really enormous body of work on breathing, he won numerous awards, including the Hodgkin-Huxley Prize, the 125th UCLA research lecture, the 2 times outstanding investigator of NIH. So without further ado, we have the Pope here to talk to you.
I don't know what to say now. Thanks very much, Fan, for inviting me. I've had a great day today. Everyone I've had a chance to meet with, it's like we didn't have enough time to talk, and I would hope we have more time to talk. I've really had a lot of fun today. And I want to talk to you today about breathing. I was fortunate enough to come here and give a seminar in September 2001, so I think it's time for an update. Those of you who might have been here in 2001, I may repeat some of the stuff that I talked about then, but we have made some advances. But we haven't solved the problem. And I think part of the reason we haven't solved the problem is that breathing may look simple, but it's not. And let me give you an example of what I'm thinking about. So here you have a simple 2-handed watch. It's an oscillator. And you could think that, well, okay, I could explain how this works by just having a spring that unwinds. And that would give me this oscillation. But it turns out that this is what the inside of this watch looks like. It doesn't look at all like a simple thing. And of course, if you have a more complicated watch, it gets even worse. And breathing has to solve many, many different things. I'll get to that in a minute. But it has certain properties that are hard to sort of merge together. It has to be robust. You're breathing from the 3rd trimester in utero to the end of life continuously. It has to be very labile. There's slow adaptations like during altitude, pregnancy, and exercise. There are fast changes in breathing like during speech, cough, and sniff. So here's some example of the changes in breathing during vocalization. And here's a recent paper by Kevin Yackel where he looks at vocalization in mice. And you see the very rapid changes in breathing. So it has to be very stable, but very labile. It also has to be variable. We think that when we're sitting here resting, nothing is bothering us. We have a very continuous breathing cycle. But in fact, much like there's heart rate variability, there's breathing variability. So if you look at an anesthetized mouse, this looks like a very regular breathing pattern with respect to frequency. But if we look at the distributions of frequency, you see that there's quite a bit of variability. So here's another boundary condition for how the system has to be organized. And it's not just about ventilation, and I'll talk about this. So nowadays, so thanks to Fan, David Kleinfeld, and others, we have this vision of the preBötzinger complex, which is the core of the breathing central pattern generator, being a master oscillator affecting all these behaviors that originate in the brainstem. And here is activity in the hippocampus of a human brain. And you can see that if you look at the EEG in the hippocampus, you see that it has a respiratory modulation. So the respiratory modulation is everywhere. this is what the system sort of looks like now. And I'm not expecting you to memorize it. I'm not going to go over it, but just to say it's extraordinarily complex. If you really want to look at the figure, we've published this. And I'm going to talk about 3 aspects of this. I'm going to talk about rhythm and pattern generation. I'm going to talk about sighing, and I'm going to talk about emotion. And I'm happy to talk more about this later or at some other time if you're interested. So with rhythm generation, the question is where, and it's this region, the preBötzinger complex. I won't go through the history about how we discovered it. If you're interested in how we named it, you can go Google Feldman BBC size, and there's a 5-minute description of how we named it. And I find it to be pretty interesting. I just don't like hearing myself repeat the story over and over again. Named after, exactly, named after a bottle of wine. The Bötzinger bottle of wine, but I'll let you go to the web and find it. So when we localize the preBötzinger complex in the human brainstem, it's about here. There's about 5,000 neurons. And the question is, how do we know the preBötzinger complex is the kernel for breathing? And there's lots of experiments that were done since our initial description, but one key prediction is that inhibiting the preBötzinger complex should stop breathing. And so here's an experiment we did where we silenced preBötzinger neurons in awake rats. And here is the breathing pattern of an awake rat. The up and down is the breathing cycle. And then we give a drug to silence the preBötzinger complex. And it takes a while for that drug to get in there. So for those of you who are curious, we used elatostatin acting on the elatostatin receptor, which were expressed in preBötzinger neurons. And that shuts these neurons off. And then after a minute in a behaving rat, they stop breathing. And in fact, we have to mechanically ventilate them because they would asphyxiate if we didn't mechanically ventilate them. So even though they're awake and know they're not breathing, they can't generate breathing movements. And then after about an hour, they're able to start breathing again. In humans, based on our identification of the neurokinin 1 receptor as a marker for preBötzinger complex, a neuropathologist at the Mayo Clinic looked at individuals who died from non-neurodegenerative causes, and he found a concentration of these neurokinin 1 neurons in this part of the medulla. And then in patients who died at the end stages of Parkinson's disease, where they have problems breathing during sleep, there's about a 60% drop of neurons in the preBötzinger complex. And individuals who died of multiple system atrophy, where there's about a 90% loss of neurons, and they have very serious problems of breathing during sleep, but they still are able to have a more or less normal problem during wakefulness. They lose almost all of these preBötzinger neurons. So this is indicative that the preBötzinger complex is critical for generating breathing rhythm. So I would check that experiment off. So the question is, if we know where, the question is how. Maybe it's pacemakers. This is an old idea. The heart is pacemakers, and anyone who knows about invertebrate studies know how important invertebrate pacemakers are in generating things like movements of the heart and the somatic gastric ganglion. It's not pacemakers. I don't have time to go into the experiments that show it's not pacemakers. I'll just stipulate it, and anyone who wants to discuss that we could talk about that later. If it's not pacemakers, maybe it's inhibition. The idea that you increase activity, you trigger a threshold, and that just inhibits it. It turns out that if you block inhibitory signaling in the preBötzinger complex, rhythm goes on in intact animals. So inhibition is important in modulating the signal. It's not critical in generating the rhythm. So the question is, what is it? And we have a new hypothesis, and I'm going to talk a bit about it. And it's a hypothesis that involves burstlets and synchronization. I want you to all take a deep breath and relax. The reason I'm asking you to do that is, one, is tie your breathing into relaxation and to realize that, in fact, breathing can affect your mental state, and also to clear your mind a little bit as I segue into the next part of the talk. So you're all familiar with this, I presume. If you're standing outside a baseball stadium, can you figure out the rules of baseball? So imagine you're standing outside the stadium. There's a game going on. You're looking at your phone, and you see the score goes up, and you hear the crowd roar. Score goes up, the crowd roars, and you propose the hypothesis. It's the roaring of the crowd that is responsible for the numbers going up, for the runs going up. Now, you realize that's ludicrous, but in fact, looking at the brain, we often think of the external things we look at as the key thing that's going on inside the brain. But now you go back to Fenway Park during COVID, and they're playing. There's no one in the stands, and you see the score keeps going up. So you realize that your hypothesis that it's the fans yelling causing that is not the case, that there's something else going on. It's similar with breathing, and let me sort of see if I can tie that analogy. So if you have the diaphragm, when it goes up, contracts, you get inspiration, you get an inspiratory burst. If you look in the preBötzinger complex, you get this bursting, and the bursting in the preBötzinger is responsible for generating this diaphragmatic activity. But you notice in the preBötzinger complex, you have this slight increase of activity beforehand, this pre-inspiratory burst. And so that's a little bit different. Maybe the pre-inspiratory burst is important in generating the rhythm, which you don't see in the output. So we did the experiment. This is now in slices of brainstem, which are capable of generating a breathing rhythm. And if you look at the hypoglossal nerve, which is the output of the slice, you see this bursting, which are inspiratory bursts. And you notice in the preBötzinger complex, there's this bursting activity, and it's 1-to-1. No surprise. But if we lower the excitability in the slice, what we find is that every time there's this high activity in preBötzinger, there's a burst of activity in the hypoglossal nerve. So that's what we expect. But you notice that in between these bursts, there's these little burstlets that don't produce output. But the burstlets look like they're rhythmic. It looks like they're occurring when you might have expected a burst. And this is actually what we see. So if we look at 2 consecutive bursts in the hypoglossal nerve, the 12th nerve, we see two bursts in preBötzinger. If we look at when they're separated, when the bursts are separated, we notice that there's a little bump in preBötzinger, but none in the hypoglossal, and it's about the time we expected a burst. Here is now a little further apart. Here again, the burstlets are when you might expect a burst, and here's even further apart. So you come to the conclusion, or we came to the conclusion, that what's rhythmogenic is not the bursting, but the burstlets. And this sort of changes your ideas around about what could be the possible mechanisms, because the level of activity is a lot less. You don't need lots of firing of action potentials. You don't need a lot of calcium entry. I mean, those of you who are cellular physiologists understand that you're now looking at a different spectrum of things that could be happening.
Sorry, I think I just missed it. What is the signal we're looking at on the top?
Oh, sure. So the signal we're looking at, I should have explained that. Thank you. So the signal we're looking at is a big fat electrode over the preBötzinger complex. So we're measuring the population activity of the whole complex, which is a few thousand neurons.
So is this like a squared, smooth, multi-unit signal?
I would say squared, but it's a smooth multi-unit signal, correct. Okay? So it's the population activity. So thank you for asking that. So the hypothesis is that instead of the burst being critical, the burstlets are critical for generating the rhythm. And that's our underlying hypothesis, which I think I'll show you even more data that supports that. So it's burstlets. What is the mechanism for burstlet rhythmogenesis that does not require pacemakers or inhibition? And our current hypothesis is that it's the network. Now, I need to segue into another experimental observation. So when we have this bursting in preBötzinger, it involves the activity of at least 1,000 to a few thousand neurons. So he asked the question, how many neurons do we need to stimulate in preBötzinger during the period between bursts to produce an ectopic burst like this? And what's the delay? Now, those of you who know the answer, don't shout it out, but those of you who don't know the answer, guess a number. And turns out that the numbers we got were surprising to us. So here's the hypoglossal nerve activity. We could stimulate as few as 3 neurons. So we used cage glutamate. We uncaged it with little dots of laser light. And if we did it for exciting only 3 neurons, we get a burst of activity. If we now add 3 more neurons, we get a burst of activity. It's a little bit quicker. And if we add 3 more neurons, it's even quicker. So we don't need to stimulate a lot of neurons to get a burst going, which was surprising. And also, look at the delay. The delay is up to several hundred milliseconds, which was surprising. Most people, many neuroscientists, would guess that it was orders of magnitude faster. But it's not. So the activity has to percolate in order to generate a burst. So how is this happening? What is happening at the network level? We hypothesize it was synchronization. And what do I mean by that? Well, let me explain this layout here. We have 3 neurons that I stipulate are rhythmogenic, going to 1 neuron that I'm going to look at. And here is the cycle, the interburst interval, the pre-inspiratory interval where this activity is ramping up in the burst. Now, if I have action potentials during this interburst interval, and they're not synchronized, they're asynchronous, the EPSPs that they're producing inside this cell don't sum up. And if this is the threshold to produce an action potential, I get no output from the cell. If now I'm starting to look a little bit later, for whatever reason, and I'll try and wave my hands about what we think that reason is, the EPSPs now begin to add up because the neurons are not necessarily firing more, but they're more synchronized. The EPSP adds up, we can get action potentials. And if we go a little bit further along, they become more synchronized, we get more action potentials. So activity can increase without requiring anything that individual cells need to do, except as a population they need to synchronize. Now, this issue of synchronization is something which is common in the physical world and the biological world. And I've seen this demonstration 1,000 times, and each time it fascinates me. So here you have several metronomes, and they're going to be asynchronous. They're loosely coupled. They're on this platform on deer cans. They synchronize. So all you need are things that are oscillators or quasi-oscillators loosely coupled. And we see this in the physical world, it's here, we see it in the biological world. Fireflies at dusk fire randomly, but very often then that randomness becomes they will synchronize their firing. So it should not be unacceptable that breathing takes advantage of this. So do we see this in preBötzinger? So here now we're looking at the population activity that I was describing before. So that's what is in this top trace. Here is a recording from an individual cell, and this is basically the power spectra. Yellow means more synchrony, and you can see here that we have a little bit of synchrony but no output. Here we get a burst preceded by this inspiratory ramp, and you see there's a significant increase in this activity indicating synchronization at these low frequencies. When we have a burstlet, that is, it doesn't produce a significant output, we get synchronization, but it's less. So it's the synchronization that starts, and then it triggers a burst where you get even more synchronization. Now, how can we test this? Well, suppose we have 2 neurons that are responsible for the output of preBötzinger. We have input from rhythmogenic neurons. If the rhythmogenic neurons are asynchronous, we expect the activity in these neurons to be uncorrelated. If the activity in these neurons is synchronous, we expect the activity to be correlated. So now I'm going to show you the activity of recording from 2 individual neurons in preBötzinger. And we're going to correlate their activity in terms of their spiking. So during this interburst interval, we have this dispersion of activity. 0 would mean that the neurons were firing at exactly the same time. This is the distribution during this interburst interval. We go into the pre-inspiratory period. You can see by eye, they're starting to now coalesce closer and closer to 0. And then if we go in the burst, they're even closer. So this is indicative that the neurons are beginning to synchronize. And this is not a case of the neurons firing more, and it's an artifact of the neurons firing more. It has nothing to do with the fact they're firing more. It has to do with their timing gets closer and closer. So we tried to model this, and we looked at a few parameters. One is we had to be able to activate a few neurons in a network. We had a network of 1,000 neurons. We had an idea what the connectivity was. It was about 1 in 6 one-way projections based on prior measurements. So that was a boundary condition. And we looked at several different network schemas, and we just ran it through a machine learning algorithm. And it turns out that the best fit we had to the boundary conditions of real data was this Enyus-Renier, which is basically a randomly connected network. Then we looked at what the optimal distribution of synaptic strengths would be. The presumption would be that any given neuron or as a population, the distribution of synaptic strengths should be normal. But it turns out that we had a much better fit when it was log normal. That is, there is a certain percentage of connections which are stronger, and that seemed to be critical for getting a reliable rhythm that a few neurons could trigger, so on and so forth. So these are explicit predictions that we're hoping now at the next stage of experimentation we can actually test. I mean, if I can twist Ed Boyden's arm enough, we might be able to get direct measurements of the degree of connectivity and ultimately whether the distribution of synaptic connections are normal or log normal. Now, all this so far has been based on in vitro work. And the constant challenge is, well, does it have any relevance to intact animals? So we recently did studies where we put multi-unit electrodes in preBötzinger to measure the electrical activity or optical fibers to measure calcium signals in preBötzinger. And when we filter this data, we can either get the extracellular spiking activity or the local field potentials. And what we find is when we look at the top here, that's the airflow. So this is an anesthetized mouse. And we're looking at the extracellular spike activity. You can see these bumps here. Now, if we had this data 10, 15 years ago, we would just say that's noise. Here are the low frequency potentials. And here again, you see the bumps. Now, when we do an analysis of the distribution of power, we see that there's considerable power at the baseline frequency, but there's considerable power at basically double the frequency. And we've controlled for all sorts of artifacts. So this is representative of a case. Now, I'll preempt the question because I didn't explain it enough. These mice are anesthetized. Anesthetized mice breathe much more slowly than a normal mouse. A normal mouse breathing at the normal frequency of about 4 to 8 per second, probably every burstlet becomes a burst. So we wouldn't see that activity in between. But when we slow the rhythm down by anesthetizing the animal, we begin to see these burstlets. So the underlying rhythm is going much faster than the output rhythm. So we're not slowing the system down by basically stretching a spring. What we're doing is we're changing the threshold and changing the threshold in a way that we're not getting a trigger at 1-to-1. We decrease the safety factor. So I think we now have the capacity to explain some of these properties of robustness, lability, and variability falls out naturally from this idea of burstlets and synchronization. We don't have to add noise to the system. To me, adding noise very often begs the question. Now, it's not saying there's no noise, but we don't need to add it. All we need to imagine is that each breath cycle is different. That is, if it's seeded by a different few neurons each time, because after each burst, each neuron is going to fire independently and not necessarily with the same timing, each breath gets seeded a little bit differently, and that could explain the variability. The lability is that this type of circuit could be pushed around very easily. But if left alone, it's extremely robust. So it solves many of these problems which have been refractory to sort of conventional modeling. Now, there's another dilemma which we have no clue as to what's happening. If you look across species, starting from our experimental mouse down to whales, there's a difference in weight. There's a difference in breathing frequency. A whale at the surface may breathe once per minute. A mouse is breathing several times per second. So there's a factor about 300 between mouse and whale. Now the question is, what's getting scaled here? When we think about many other processes in the brain, like LTP and so on, we think of it as the same scale, that a whale or an elephant or a human is going to be in the same time scale as a mouse. Yet the mouse is breathing 4 per second. We're breathing every 5 seconds or so. So what's getting scaled? We don't know. And we hope that by the time we can figure out what's happening in mice, we can begin to specifically investigate what may be changing by techniques that would allow us to do experiments in these other species. So I need you sort of reset your brains. Now, when we think of rhythm, for breathing, we think of inspiration, expiration. And we originally thought that the preBötzinger complex was generating both phases. Just like the day and night comes from the rotation of the Earth. There's one thing that's oscillating, you get two distinct phases. We thought that that's what was happening with preBötzinger. And so the way we think about breathing was that there was this quote from the New York Times, and since the way she breathes, exhalation following inhalation, has already gotten on your nerves after just 24 hours. But the notion that this is inexorable sequence of inspiration, expiration, inspiration, expiration, it reinforces the idea that it may be coming from one place. But here's some experimental data. So here is, at the top trace is airflow in a young rat, and down is expiration, up is inspiration, and we have an expiratory muscle on the bottom. So this looks pretty boring. The way this animal breathes, it has a little exhalation, then an inhalation. It's very regular. Now, if we give this rat a little bit of fentanyl, breathing slows down. But watch how it slows down. I have sound on this one. Expiration, inspiration. Now watch this breath. Expiration, no inspiration. You notice the expiratory muscle keeps going. Now, when this happened in the lab, we didn't believe it. I mean, how could this be? Now, there could be a simple explanation. You're just periodically blocking the expiratory output. But turns out that through many experiments, we found that there's a second oscillator. And the second oscillator is a little bit more rostral. It's around the facial nucleus. And the reason we missed it was that we neglected to consider the fact that in the experimental animals we were looking at, expiration is passive. So at rest now, your expiration is passive. You inhale and you passively exhale. You just let everything that's elastic come back. So there's no active expiration. But when I speak, I have to generate significant airflow over my vocal cords. There's active expiration. When I increase my ventilation significantly when I exercise, I got to turn on expiration. And that looks like it's a second oscillator. And I'll show you data that I think is pretty convincing, but there's lots more experimentation. So here's a rat. The top trace is their inspiratory activity. I think it's the diaphragm. The bottom trace is their abdominal activity. And you notice there's no abdominal activity. There's plenty of inspiratory activity. We put channel rhodopsin, thank you, Ed, into the region of the expiratory oscillator. When we turn the laser light on, look what happens. The animal starts generating significant expiratory activity. And when we turn the light off, it goes away. And there's not much change in inspiration. So this and a lot of other pieces of data suggest that we have two oscillators, one for inspiration, one for expiration. We can even put mice in a state that they're no longer inspiratory breathers, they're expiratory breathers. But that's not the way it normally is. We're inspiratory breathers because we have a diaphragm. Amphibians and reptiles don't have a diaphragm, they're expiratory breathers. So let me sort of elaborate on this. So I have separate oscillators. So if we look at non-mammalian vertebrates, they don't have any diaphragm. Their lungs are simple. That is, the surface area of their lungs is relatively modest compared to the volume of their chest. And they don't have a large surface area. They're cold-blooded because they can't exchange very much oxygen. And they have smaller brains, which require lots of oxygen. They don't need powerful inspiratory efforts because the way they breathe is they exhale and relax. So they have an oscillator that drives expiratory-dominated breathing pattern. That's what we think is the expiratory oscillator. Then evolution comes along. We invent the diaphragm. We get complex lungs. The surface area inside our 3-liter chest is about 70 square meters. That's about a third the size of a tennis court. That's a lot of squeezing in. The alveoli are very small, but we can get lots of oxygen in because it's 70 square meters. It allows us to be warm-blooded and to have bigger brains. So powerful inspiratory efforts are needed and possible. And so a new oscillator, the preBötzinger complex, to drive the diaphragm that couples to the old expiratory oscillator. And the coupling normally is so strong that you would never think that they were independent. Okay, I want to now segue into sighing. And this ties into a little bit about our lungs and what's weird, what's special about our lungs. So you all know what a sigh is. It's a deep breath. Now, why do we sigh? Well, if we look at the lung, the gas exchanging surface of the lung are these little tiny spheres called alveoli, and they're tiny. They're 1/10 of a millimeter across, 100 microns. Now, that gives us a large surface area because there are 450 million of them, which gives us 70 square meters. But there's a problem. The problem is they're so small and they're fluid lined with this stuff called surfactant. They have a tendency to spontaneously collapse. Not very rapidly, but they collapse. And when they collapse, they don't disconnect from the airway. It's a bad animation. But when they collapse, there's no gas exchange. And so that piece of the lung is useless. Now, if that spreads over time, it can be very bad. And I'll explain an example of that. So a normal breath is not enough to pop it open. If you take a balloon, you make it wet on the inside, and you collapse it and you try and blow it up, it's not going to be easy. You're going to have to put an extra effort to blow it up because the surface tension of the water is keeping the sides of the balloon together. And so what you need to do is you need a big breath, and then that'll pop it open. So you're sighing every 5 minutes. You don't believe me? Lie down someplace quiet, pay attention to your breathing, and you'll notice every couple of minutes you're taking a particularly deep breath. The process of self-discovery for me was pretty amazing when I actually did it. And I said, oh my goodness, I actually am sighing every couple of minutes. In the days when iron lungs were used to treat individuals who had polio and couldn't generate sufficient breathing movements, iron lungs were used, and iron lungs were used initially where the volume of the breath that was generated was the normal volume. For adult males, it's about 500 milliliters. they had a high mortality rate. Why did they have the high mortality rate? Well, you may have already figured that out. Their lungs were collapsing and there was no countervening force. They initially tried to just increase the volume. That lowered the mortality. But then someone tried, let's give a big breath every once in a while. And that was sufficient. Now, if you go see someone being mechanically ventilated in the hospital, you'll notice that the pump is like this, and then every couple of minutes there's a big breath. And that's the mechanical equivalent of a sigh. Now, we got into sighs because someone in my lab, Viktor Janishevsky, noticed that when animals are particularly stressed, their sigh rate goes up and they release bombesin-related peptides. And we said, maybe they're related. Bombesin was cheap, and we just decided to, well, let's inject it. So we injected it in the preBötzinger complex and predicted that maybe it would increase the sigh rate. And that blocking the receptors in preBötzinger should lower the sigh rate. So we did the experiment. So the bombesin-related receptors, peptides, are normetin B and gastrin-related peptides, NMB and GRP. So if we inject them into preBötzinger, here's 5 different rats. This is rats. And each tick here is a sigh. So this is 1 rat, 2 rats, 3 rats. When we injected the peptides, you notice there's a significant increase in the rate of sighing. When we block the receptors, you notice the sigh rate goes down to 0. So it suggests that there's a circuit that involves these neurons that are releasing the peptide to neurons that have the receptor for the peptide in preBötzinger. It turns out there are populations releasing this peptide around the facial nucleus, the same place that we found this expiratory oscillator. There's another function there of neurons that are releasing these peptides to produce sighing. And so we came up with a story that you have in the parafacial region, neurons that are releasing GRP and NMB, and if you knock these neurons out, the sigh rate goes way down. And if you block the peptides here, the sigh rate goes way down. And the thing is, these are probably not output neurons. They go through neurons that have the marker somatostatin producing sighs. So we have a circuit for production of at least the sighs that seem to be responsible for regulating lung health. The sighs that come along with when you're stressed or when you're joyful and you're are probably due to release from the hypothalamus of these peptides. Now I want to talk about breathing and emotion. And this is something which is becoming more and more intriguing to neuroscientists. Nature Reviews was kind enough to put a cover of a meditating mouse with respect to a review that we wrote. And we wrote a paper that went viral. The New York Times wrote about it. And I have to say, one advantage of this paper going viral is that a colleague in the Department of Psychiatry at UCLA who doesn't read the journal the paper was published in, but reads the New York Times. And she was maybe 100 meters from my office. And we've started a collaboration to try and put together work in animal models and humans. So if we have eye function modulation of breathing, we have volitional modulation of breathing, as you can see me talking here or breath holding. We have a motive control of breathing. Now, how do we know that they're separate? And I think the prime example is locked-in syndrome. Locked-in syndrome follows a devastating stroke or injury in the ventral brainstem that is going to interfere with the sending signals coming from the cortex for all volitional movement caudal to the lesion. So these individuals in the extreme can blink and move their eyes laterally, but they can't move anything else. If they're ventilated, they will still live. Their cognitive function is perfectly normal. And we had colleagues who were studying a locked-in patient who had a a locked-in patient who had a caretaker, and the patient blinked to the caretaker so he could communicate to the caretaker. So let me directly quote from the paper. Although the subject was seen to have no voluntary control over his respiration, that the subject was asked, hold your breath, breathe faster, no change, very robotic breathing. He was witnessed to laugh in response to humorous remarks. What happened was the breathing didn't change, and the subject, the patient, was responsive to hypoxia and hypocapnia, so responded to changes in blood gases, but no volitional thing. Now, breathing is intact because those structures, like the preBötzinger complex, is caudal to the lesion, and the connections from preBötzinger complex going down the spinal cord to the diaphragm and other muscles is intact. Where that gets interrupted is when individuals have neck injuries, and then they stop breathing, not because the preBötzinger complex stops, it's because the connectivity is broken. But getting back to this, the patient suddenly, the breathing problem suddenly changed. And they said, what happened? And the patient said, you told a joke and I laughed. And every time they told a joke the patient found funny, had this very stereotyped breathing pattern, which we know for laughter is we inhale, ha, 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 ha. And in the neuroscience community, we have several individuals who have highly distinctive laughs. I won't name names. And it's emotive. It's not volitional. It's emotive and can be highly idiosyncratic. So you have this emotive control of breathing that's distinct from the volitional control of breathing. Now, what about going in the other direction? What about the effect of breathing on emotion? And here, to understand what likely is going on, we need to think about oscillations in the brain. We know that there are all sorts of oscillations in the brain over a broad range of frequencies. What are these oscillations are doing? Well, these are speculation by other people. There's binding. You have signals that relate to different sensory modalities. Excuse me, you need to pull them together. And timing is very important. So auditory and visual signals get pulled together. You see me as one thing, hopefully, not as two different channels. But you know when you're watching a TV program or a movie and the sound gets desynchronized, it gets delayed by a few milliseconds, your brain has a hard time putting it together. You feel very uncomfortable. And probably these oscillations are helping your brain figure out when things should be tied together and when they could be unrelated. So it's an important role of oscillations. They increase reliability of transmission. That is, if here is threshold and I have a signal that's kind of a tonic right around threshold, it may fail. It may fail to get across because it may fail because of noise to get above the threshold. If you have an oscillation, you're guaranteed to get above it. And so these oscillations seem to be very important in assuring signal processing. So what about breathing? It's slower at rest than these other frequencies in humans. In mice, delta is about in the range of breathing. So delta and breathing in mice can be interacting, but in humans it's slower. It's under volitional control, and to the degree that it could be involved in signal processing in the brain, we can control that, whereas most of us, I don't think, can control the other oscillations. There may be individuals like yogis who can control them, but they're exceptions. So breathing signals are everywhere behaviorally. So your pupil oscillates with the respiratory cycle. So the top is the phrenic nerve going to the diaphragm. The bottom is your pupil diameter. It's a small oscillation, but it's real. Don't ask me why. This is just observation. The reaction time is breathing modulated. That is, your ability to react to something which requires a reaction varies between inspiration and expiration. Martial artists and boxers will pay attention to that when their opponent is inhaling or exhaling because it will affect their ability to respond to an aggressive action. Fear, not surprise reaction is breathing modulated. So Christine Zolano showed individuals normal faces and fearful faces. And they responded to the fearful faces, but the fearful face response was modulated by whether they were inspiring or expiring. It's a couple of milliseconds, but it's a real difference. Whereas if it was simply a surprise, no difference. So breathing rhythms are everywhere in the brain. You see them in rodents, in many different parts of the brain. So you have olfactory ball of hippocampus, somatosensory barrel cortex, prefrontal cortex. It's basically everywhere. And it's hard to think that this doesn't play a role, particularly if I go from breathing, the mouse breathing at one frequency to another frequency. Now, mice don't change their frequency volitionally. They don't meditate, but we'll get to that in a minute. But we also see that in humans. So if you look at the hippocampus has a respiratory modulated frequency. And so if I slowed it down from 12 per minute to 5 per minute, maybe it changes. We know a single breath is enough to make you feel calmer. And breath work has been established, probably for thousands of years, as having a profound effect on emotional and cognitive state. So how does breathing affect emotional state? So we have the preBötzinger complex. And we did a study with Stanford colleagues Mark Krasnow, Peng Li, and Kevin Yackel, where we looked at projections from preBötzinger complex to locus coeruleus. And what we found is that when we killed the cells that had this projection by doing some molecular magic that Luqian Lu helped us with. Didn't help us with, he actually did. What we found was, if under normal circumstances, this is the distribution of breathing patterns, when we lesion these cells that projected from preBötzinger complex to locus coeruleus, the breathing rate slowed down. And when we looked at their behavior, they became less active, groomed more, and when John Huguenard looked at their EEGs, it was very convincing that the animals were calmer. So we built a Just So story, and we were able to get it published, that this is one of the ways breathing can affect emotional state. So what are the other ways? Well, we can have direct effects. We know that there are direct projections from preBötzinger to all sorts of places. And by the way, this spot I have emotion, I don't mean to say that that's the localized center for emotion in the whole brain. It's just a surrogate for various places in the brain that can affect emotional state. So here are projections from preBötzinger complex. The green is direct projections, and this is a rodent brainstem. And then if you go through locus coeruleus, it goes through everywhere. So there's a way for signals directly from preBötzinger to get throughout the entire brain. Now, breathing affects emotion. If I tell you to breathe slower and it makes you calmer, it could be a placebo effect. So we decided to see if we could study this in mice. I know it sounds crazy, but we were able to convince NCCIH to give us a little money to try this. And it took 3 years to figure out a way we could get mice to slow their breathing pattern. And so what we did is we had control mice. And so what we did is we put an opsin into preBötzinger complex in the inhibitory neurons in preBötzinger complex. In the control animals, we injected the same virus without the opsin. And we did the same manipulation. And here I'm showing a superposition of many breathing cycles triggered off this peak. So this is before and this is after. And you can see there's no change when we shine the light. If we looked at doing this 30 minutes a day for 4 weeks, and we looked at their fear response, and we, as Michael Fanzelo, who's a world expert in this is the range of fear responses, freezing responses we saw, and this is in the normal range. However, in the test animals, we did the same thing, but in the test animals, they had the opsin in the inhibitory neurons. You see, every time we stimulate, we get a lengthened expiratory period, so that slows their breathing rhythm down, 30 minutes a day for 4 weeks. Question is, does it affect their behavioral response? And when we did a test of their freezing behavior, you can see that there's a significant difference at a very high probability. So this is a bona fide effect, and we think that it has has value for several reasons. One is that it's not a placebo effect. Okay, I would say mice don't believe in placebos, and moreover, we repeated this experiment with a different group of experimenters. So we tried to control for the possibility there was experimental bias, and I think we've controlled for that. And it's now a platform for studying mechanisms. That is, for 3 years I've been talking about this, and people have been asking, well, how do you do it? And I've refrained from telling them because I think the postdoc who did it, Raquel Abreu, I need to make sure she got full credit for it. And I wanted to make sure that we got the publication out before other people started to replicate this. And now I think other people will replicate it for whatever studies they're interested in, including pain, Fan. In other words, does the animals that go through this process, are they responsive to the standard metrics that neuroscientists use to measure response to pain? My prediction is...
Did you measure freezing during the freezing? Do you see a difference?
No, we didn't measure it. We didn't measure it. That's a good question. There's 1,000 things we should have done. These are hard experiments, and the first 2.5 years, everything failed. So we were just anxious to sort of push it along. It's a demonstration of that it can work. So we have a conjecture about why breathing evolved to affect emotional state, and we think it's sighing. That is, mammals develop these lungs, they needed to sigh. For some reason, sighing became calm. It was calming. There's no a priori reason to explain that, but that's the case. And I think if you could take an animal could take advantage of that. A predator is frightened, tries to hide. If it sighs, the hiding may be more effective. They may be a little bit calmer, may give them some selective advantage. Now, I'm not saying this is a hard demonstration. It's a late night conjecture. But that sigh, and then the brain eventually hijacked the breathing signal. And for us now, we can now hack into that for advantage. And I have drunk the Kool-Aid. I really do think that breath work can have positive effects. A recent meta-study of 3,000 papers related to breath work threw out all but 70 of them as being underpowered and poorly controlled. But the 70 that they found were valid came to the conclusion that 5 minutes a day for a month produces significant and measurable changes in affect and in physiological variables. So I would say try it. So this is what I just said. So here we are back to where we were before. And I talked about rhythm generation, sighing, and emotion. There's a lot more going on. And unfortunately, I would need a whole course to teach about it. So with respect to rhythm generation, burstlets, synchronization, Erdős-Rényi, log normal synaptic strength. That's the takeaway there. For size, Bombesin peptide microcircuit underlies the effect of breathing to locomotion. And as far as emotion goes, breathing does affect signal processing in the brain. So with that, I, of course, like to thank the agencies that have funded me and all the great people who did all the work I talked about. Thanks very much, Fan, and I'm happy to answer questions. Brad? Brady?
On the rhythm generation side, is it all synapses or are there also gap junctions?
We've looked at gap junctions, and we're convinced they exist, but they're not critical. I can't say much more at this point, and part of the problem is most of the blockers of gap junctions also have other effects, and you can't disentangle the other effects from effects on breathing. When we did pairwise recording, we found that, in fact, there was a electrotonic coupling between cells, but it was pretty weak and could not account for the dynamics that we see. Yes?
Back to the difference between reptiles and mammals. Have you done experiments to try and find inspiratory pacemakers in reptiles, or were they absent?
I've tried to get other people to do it. Okay, but unlike Tom Sawyer getting people to paint the fence, I have been unable to do that. But there's no evidence yet of anything like the preBötzinger complex or its precursors in reptiles and amphibians. So if I would just say we have an absence of data, but I think that there's highly unlikely based on the anecdotal interpretation of that data. Yes?
I'm just curious from a surrogate perspective if you have any idea in the locked-in patient how they would have the motor breathing changes but not the volition.
Well, I think that it's a simple neuroanatomical explanation. That is, the pathways that are responsible for the volitional control are running down the ventral part of the brainstem. And that's where the locked-in stroke or lesion is occurring. Whereas the ones controlling emotion bypass that, which suggests that, of course, they're going more dorsal. But the exact pathway is, I don't know. But I don't think this is quite a hand-wave explanation. I think that's has a real basis in the neuroanatomy. Yes?
So I had a question about the modulation of the frequency of breathing, the period. And you mentioned that in anesthesia, it looks like instead of changing the fundamental frequency, there's skipping, basically, right? But there must also be, because if that's the only mechanism for changing the frequency of breathing, it would suggest that the changes would be quantized. But I assume that there are other mechanisms that,
It's a wonderful question. We think now that there's two flavors of modulation. There's continuous, which is what you're describing, which I think for us, up until we made those observations in vitro, we would have totally subscribed to. And so I think there's continuous modulation of breathing. But we have some data now from this that there could be sort of quantal changes in breathing. And where we're thinking about this happening is with sniffing. So with sniffing, there's an extremely rapid onset of sniffing. When it's not as if you go from, a mouse goes from 4 hertz to 12 hertz over a period of several seconds. It does it instantly, milliseconds. And we think one of the ways that that's happening is that the mouse is in an environment where it thinks it may need to sniff. It's energetically costly to sniff. It may expose the animal to making noise. And it's prepared then to turn on the sniff very quickly because it has these high-frequency burstlets that you don't see in the output. So it's still breathing at the normal frequency, but it's producing these burstlets. And then as soon as it maybe gets the first odorant molecule, it's very close to threshold and immediately can begin sniffing. So we think that in the real world, and perhaps maybe in vocalization, that there's this utilization of throwing in these extra burstlets. So in fact, when you have to extremely rapidly change your breathing pattern, you're not required to ramp things up and ramp things down. Take a look at your heart. When you go and exercise, your heart rate doesn't go from baseline immediately up to double baseline. It takes a while to ramp up. Whereas breathing, you can double your breathing frequency just like that. So there has to be some way to rapidly do that. And you could say, well, all you're doing is just driving some excitatory input, and that's sufficient. And that may indeed be the case. But if a much faster way is to prime the circuit, and then you can trigger it like that. It's pure speculation. Yes?
You mentioned it's really hard to train or get rodents to slow down their breathing. I assume you try to reward them or to train them in some way.
Well, other people have used sort of operant conditioning, and they get a little bit of slowdown. Here we get a we can have basically an arbitrary slowdown. But we're limited by our perception of whether the mouse tolerates it or not. So we start out in the training with a relatively brief pulse, about 200 milliseconds, and we can extend it up to about 750 milliseconds. We don't go further than that. Yes, we threw as many things as you can imagine up against the wall, and some more than even you could imagine. And we didn't get anything that was reliable or reproducible till we did this.
Okay, so it is possible to reduce it a little bit with condition.
A little bit, a little bit.
I was going to ask if that were not the case, you think they don't have volitional control of breathing?
Oh, they definitely have volitional control of breathing. But don't anthropomorphize. Okay, if you anthropomorphize, then maybe they don't. But I think there's definitely all the circuitry there, and you can. I would imagine that you can get them to respond in a way that's volitional to something. But to be candid, we don't work on that aspect of it, so I don't really have any expertise there. You had a question in the back?
Yeah, for your field potential recording, we did a bath, we dosed with fentanyl. Do you think that it's silencing the neurons, or is it just disrupting the state of this environment so that you can no longer see it with field potential?
So what's happening when you record in preBötzinger, you see that the neurons themselves are a little bit hyperpolarized. And that could be sufficient that when they're supposed to generate a burst, they generate a burstlet, but it doesn't reach threshold. We know that there are opiate receptors in preBötzinger. In fact, we are working on trying to understand how fentanyl is causing apnea that leads to fatal overdose. And we think that a large component of that is the fentanyl acting in preBötzinger, which has nothing to do with the fentanyl or other opiates acting as analgesics. So we're trying to bifurcate that problem. But we think what's happening is we're increasing the threshold for the conversion from burstlets to burst.
So, have you also looked at input from HCM receptors on that?
No.
Jack,
We should.
There is an expiratory rhythm generator. How, fentanyl obviously causes overdose, supplies the inspiration oscillator. Why can't the, I assume that when you're young, the expiratory oscillator is continuous active, where as you become adult, this becomes a conditional, not always good. Is that the idea?
You're so astute. You answered your own question. So,
But what the mechanism?
well, okay. So here's a dilemma. In mammalian births, and certainly in human births, there's a surge of endogenous opiates in the mother. Now, if the preBötzinger complex is sensitive to opiates, you might expect there'll be a depression of inspiration. As far as we can tell, this region which has the expiratory oscillator doesn't have opiate receptors. And so what we think is happening at birth is that this is driving the extra excitability that you need to prevent the apnea from the endogenous opiates getting into the fetus through the maternal circulation. And it actually takes a couple of days for it to clear out. And I think after that point, there's a waning of the effectiveness of the expiratory oscillator if the inspiratory oscillator fails. And we're just able to artificially separate the two. But we think that in normal mammals, it just fades away. Maybe because it's not needed in an adult. Yes?
Is there a relationship between yawns and size? Is there a connection there?
I wish I had the answer for that. There's lots of ideas about why we yawn. It goes across the gamut that's related to blood gases, to I'm bored and I want to communicate to you I'm bored. A lot of human primate communication is facial expression. And so if I'm yawning, you're getting a signal that I may be bored, and that may be part of the yawning reflex. The mechanical aspects, the implementation of a yawn is sequentially different than a sigh. So I would say that there may be some reconfiguration of the circuit. We have, it's not just yawning, it's coughing, sneezing. They're all using the same musculature. And then you have things where you have to modulate breathing, like swallowing and chewing. And so that's where the system that's why it looks like that inside of a watch. And we just haven't been able to disentangle it. I mean, it's a great question. And quite frankly, and a fan asked me about hiccups. NIH is not going to pay me to look at yawning. If you know some wealthy donor who wants to pay for that, maybe we look at it. But as a matter of fact, it's not just a curiosity. We have to sort of find things that we can get someone else to pay for. And yawning doesn't fit into that category, unfortunately.